Zebrafish development begins when sperm travels to the egg and forms the zygote. This marks fertilization. Entry of the sperm causes a reaction called the cortical reaction. This is a wave of calcium released across the zygote. Then accumulation of cytoplasm at the animal pole occurs, forming a structure called the blastodisc. This is mediated by the pulling of actin filaments away from the vegetal pole. Zebrafish embryo contains a high volume of yolk, which works to inhibit cleavage. Therefore, cell division occurs at the yolk-limited site. The blastodisc undergoes meroblastic cleavage at the vertical plane, which produces two blastomeres, which remain attached to the yolk. The two blastomeres are cleaved at right angles to produce four blastomeres, which still remain attached to the yolk. The four blastomeres is again cleaved, this time in two vertical planes, and produces 8 blastomeres. The 8 then becomes 16. This marks the point where, for the first time, some blastomeres are completely cleaved from the yolk. 4 central cells are also seen fully surrounded by marginal blastomeres. The 16 then becomes 32. The 32, for the first time, are cleaved at a horizontal plane to produce 64. As the number of blastomeres continues to multiply, it results into some blastomeres being buried by the uppermost layer of blastomeres, or the enveloping layer. This produces the first deep cells. From this point on, cleavage becomes to be irregular and are asynchronous, creating a ball-like structure on the animal pole called a blastula. The number of deep cells beneath the enveloping layer continues to multiply. At 512 blastomeres, the yolk syncytial ring forms around the blastodisc margin, which moves away from the blastodisc, decreasing the size of the embryo. An internal and external yolk syncytial layer is produced and begins to swell, marking the beginning of epiboly. During epiboly, uppermost cells travel to the sides of the embryo and encloses towards the vegetal pole. This causes the blastoderm to enclose around the yolk. At the gastrula period, multiple cell movements occur such as involution, convergence, and extension. At the shield stage, cells continues to move around. Deep layer cells meet towards the germ ring and form the embryonic shield. At the embryonic shield stage, cells form the axial hypoblast, which is the external layer of embryo. The hypoblast moves towards the animal pole. Axial mesoblastoderm reaches the dorsal side. At the bud stage, epiboly and gastrulation is complete. Tail bud is formed, and the head region at the animal pole is formed. The embryo then undergoes segmentation, which involves various morphological factors that gives rise to the fish's morphology. Segmentation is when organs become visible. Somatogenesis begins with mesodermal walls called somites. Somites give rise to skeletal muscles, smooth muscles, and vertebrae. The formation of somites is established using an interaction of morphogens called the wavefront and clock model. This model produces efferent B at the posterior end of the somite and efferent A at the anterior end. Electrostatic interactions repel the forming somites, enabling individual muscle and bone formation. Neurulation also commences, which forms neurons and cranial vesicles, which develops as the brain. Then commences the frangula and hatching stage. During the frangula stage, the fish develops structures such as the fins, mandibula, and gills. It also increases in length and pigmentation begins. The hatching stage then occurs, where the circulatory system is formed and the heart begins to beat. The head compacts and the tail begins to hatch away from the yolk. Final morphogenic changes occurs in the fish as hatching finalizes the completion of embryogenesis.